Yes, I should have been recording this. I apologize. I don't know why it's not recording. So at this point, I'm not going to go back. It's not, for some reason, someone asked me to switch color, and now it's not working. So you see here, you see OH next to each other. You assume that OH has, why is there OH? Why not just O? Because oxygen needs two bonds. You see one bond here is to the oxygen, is to the hydrogen, the other bond's to phosphorus. This bond is here to phosphorus, one to hydrogen, one to phosphorus, and then this one has two bonds to phosphorus. Why does it only have one? Because it needs two bonds to have a full octet. Phosphate has a charge, it has a negative charge. When it's bonded to glycerol, we draw this like this, and you have to understand there's a molecule that you should get, if you want to be comfortable in this discussion, you should get used to this little drawing right here. This little alcohol. Why am I drawing four little lines? So all that was correct. So why does I was going to try to copy the picture, but it didn't work. So we'll just let it go. I'd have to open up that. It just would be too annoying. I know that each carbon is going to have four bonds because carbon makes four bonds, period. As your generation would say, period. Four bonds. How do I know that? Carbon is in the fourth period. Fourth. Not including the transitional elements. Huh? I'm sorry, group. It's in group 4. Thank you. Carbon's in group 4, not including, if you count them from left to right, be 1, 2, 3, 4. But we're not including the transition elements. Is that clear? Transition elements are filling d orbitals, and they do all kinds of interesting things. But carbon is the fourth group from the left, not including transition elements, so it makes four bonds. How do I know that? It's on the fourth group. So here we have 3 hydroxides. This molecule is an alcohol. Anytime you have, I don't know if you learned that in chemistry, but anytime you have OH groups on something, it's called an alcohol. You're used to ethanol, which is a type of alcohol that has 2 carbons, right? This is an alcohol that's known as glycerol. The reason I'm asking you to know this is because it's involved in a lot of different biochemical pathways. It would be good if you knew it. For instance, I can take this molecule of 3 carbons with its 3 OHs. OHs are amazing little structures. By the way, OH is a group. It's a functional group. It's one of the functional groups you should know. It's one of those things you should know that's called hydroxide. By gosh, you should know hydroxide. There should be never a question, hydroxide. OH, hydroxide. If you have OHs, it's an alcohol. If it's 3 carbons with 3 OHs, 3 oxygens, it's a glycerol. Yes, sugars are a type of alcohol. Yes. That's how it's so easy to turn sugar into alcohol, because in a way, technically, huh? I can't read. It's not it's not an alcohol, but it's if you go by the definition with anything with an OH group on it is an alcohol, then sugar's an alcohol because it has OH groups in it, right? It's not, right? There's a reason it's not. We'll talk about it later. But if we just use that definition, yes, I guess you could classify it as that, but we don't call it an alcohol. There are certain qualities of alcohols that it does not have. Is that clear? But it's easy to change it into an alcohol. It's easy to take fruit and grapes, grapefruit, any fruit, any vegetable, anything with sugar, and turn it into an alcohol. It's easy. It's called fermentation. Easy. Bacteria do it all the time. So it's alcohol fermentation. Yeast do it. We use beer's yeast to make beer, and we use other yeast to make other alcohols. But all the yeast do is break it up, use the sugar, and produce alcohol as a byproduct. They produce something called ethanol, which is the stuff that we drink and we also put in our cars to burn. If you drink alcohol, that's what you drink, ethanol. So anyways, you got these three alcohols. What's interesting, three hydroxides. What's interesting is you can take this chain of carbons. Take this bunch of carbons. Let me get a picture because I'm not drawing all this. Give me a second. What is interesting here is that I can take this chain of carbons. Look at it. Look at it. It is a chain of carbons. That's all it is. It would normally have an OH group at the end, and then it has this long chain of carbons. You call these fatty acids, and these could be saturated or unsaturated. These are saturated fatty acids. They're small fatty acids. Some of the chains are very long, 20 carbons long. These are obviously short, 1, 2, 3, 4, 5. And the length of the chain tells you the type of fatty acid. And each fatty acid has a different purpose in your body. There's an oleic acid. If you've ever heard of oleic acid, there's a really great movie about a kid who couldn't digest oleic acid and went from being a normal everyday kid to being someone who's bedridden. And it was their parents got together and figured out how to solve the problem by changing and help him deal with oleic acid. Unfortunately, it didn't help him in the long run. Oleic acid. It's a type of fatty acid. There's a lot of different fatty acids. Fatty acids have a carboxyl group. That's one of those functional groups. So hydroxide's one. Another one is C double bond O, OH, connected to something. This is a carboxyl group, also known as a carboxylic acid. Fatty acids are acid because of the carboxylic acid group. This makes it an acid. Carboxylic acid makes it an acid. It's a fat because of the long chain of carbons. Anything that's a long chain of carbons is hydrophobic. I'm sorry. It's afraid or hates water. So it's going to not like water because it's a long fat. That's what fat is. It doesn't mix well with water. It does not go into solution with water easily. Oils, all oils are forms of lipids. These are lipids are generally long. Another word for lipids, I'm going to put the word lipids. There's several different kinds. There's fatty acids that have an acid group. Not all lipids have acid group. There are steroids. And another word for lipids and fatty acids, a kind of a big umbrella group, is hydrocarbons. Why? Because you have hydrogen. They're mainly made of hydrogen and carbon. That's most of the molecules made of hydrogen and carbon. We call them hydrocarbons. What do you think we call gasoline? What kind of fuel is gasoline? What kind of fuel is ethanol? What kind of fuel is, well, not ethanol, but oil? What kind of fuel is any of these fossil fuels? A lot of them are called hydrocarbons because they're long chains of carbon and hydrogen that we burn to produce CO2. So, that's a lot. But so far, let's go back and name and number them. 1, we have hydroxyl group. 2, we have a carboxyl group. 2 functional groups have been introduced to you. 1 large molecule type, glycerol, that you should know. Why is glycerol such an important little molecule? Because it's used to connect fatty acids to. Fatty acids. When you go into the doctors and they do a blood test and they test your tricarboxylic acids, not tricarboxylic, what am I saying? They test for triglycerides. They're looking for these 3 fatty acids attached to a glycerol. It's floating around in your blood. Not loose, it's in a ball of lipids. And we look to see how much cholesterol is in there. And if you have a lot of the HDL, high density lipids, that turns out to be, I think, good. And high LDL turns out to be bad. One form of these balls of lipids creates artery disease, and another part does not. So it's important that you measure these moving through your lives. All right. So what's interesting is this glycerol molecule got turned into this by taking this fatty acid, which again, I'm going to draw it out. It's a carbon, double bond O, OH. Then, do I need to put another bond on that? Carbon? No. There's 4 bonds. We're done. Does oxygen have all its bonds? Does oxygen have all its bonds? Yeah, 1, 2. Does this oxygen have all its bonds? Yeah, 1, 2. Right? Everybody's happy here. So this has to be connected to something, though. What's it connected to? Another carbon, which is going to have 2 hydrogens. Then it's going to connect to another carbon. Then 2 hydrogens. Then another carbon. 2 hydrogens. Another carbon. 2 hydrogens. Another carbon. 2 hydrogens. Another carbon. Then at the end is going to be how many carbons? The 2 hydrogens. How many hydrogens? There's 2 hydrogens. And then what now? It's the end. What do? Another hydrogen. I can't, if you put oxygen, you'd have to put a hydroxide. You have to put an OH. You couldn't put just an O because then oxygen would need another bond. So that completes all the bonds. All the bonds are nice and happy. Do you all agree with this? So when I look at this, I can see that all the bonds on this are absolutely accounted for. Do you agree? Okay. So now I have glycerol. Here I have a glycerol molecule, carbon, connected to carbon, has hydrogen, 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 has 3 OH groups connected to it, and hydrogen's at the end. There's my glycerol molecules. We make this a lot. So now you get this fatty acid coming close. What do you think is going to happen here? Who can tell me what's going to happen? Look at that and tell me what's going to happen. Say what? So the O here is going to do what? We want to connect these two. Can I just stick it on here?
Oh, the double bond will break.
Double bond's going to break, and then what's going to happen?
And then it will connect to the OH for another bond.
So then what's going to happen is that this O and this H, that was messy. Sorry about that.
It can bind to the O because those are partially
That's exactly right. It's dehydration synthesis. So this oxygen and this H are out of there, and they form what? What are they going to form? This O and this H are going to come out and form what? Well, it'll form water.
Okay, how it makes, where it binds to the H is already binding to the O, and then.
That's right. I'm sorry. Yeah.
So that H is gonna bind to the O, and then the O is already attached, the O next.
So that wouldn't work, right? Do you agree that that won't work? Because that doesn't form water, right? Does everybody agree that that won't, what I just drew on the board does not work?
Yeah.
Do you see what the look, guys, you have to look at the board. You can't be looking off to the side. If you're not looking at the board, you're not understanding. Do you see why that H and that O leaving would only form, somebody said hydroxide? That does not form water. It has to form water. So how can we combine these two to form water? We're not going to add hydrogen. I heard someone say that. That was a good guess, though.
Would that hydrogen bind to the oxygen?
Oh, I hate when it does that. And I can't undo. Why can't I undo? That makes no sense. It didn't show. Now I can, but I couldn't. What? This is a double bond O. What can happen? I need to get one oxygen and two hydrogens out of there to make water. My objective is to make water and connect these two. It's like Lego sets. How do I get rid of the O, one O and two H's to make a connection between these two? I need to make a connection between this group here and this long chain of carbons. Carbon can only form 4 bonds, and oxygen can only form 2. Yeah.
I know how the end result would look like, but I don't know how you would dehydrate it. Like I know, because the carbon on top is going to form with the carbon below, right?
Right, but there has to be some. Yeah.
What if we remove the OH and the H there?
What OH? I mean, this OH here. So you want me to get rid of this OH here?
No, not that.
Not that one. Okay, which one?
The one on top.
So this OH here.
Yeah, and that H.
And this H here. You want these to leave? Yes, that would make water. You could also get rid of the other OH. It'd still be the same. So you get rid of two of those H's in an OH group, or this OH group, and that I'm honestly not even sure which one you would get rid of, but you would end up with a carbon connected to a carbon, connected to a carbon, hydrogen, hydroxide, correct?
Right, because you removed that.
Hydrogen, still hydroxide here, right? But now here's a hydrogen, but now, what am I connecting to that carbon?
Oxygen.
There's going to be an oxygen that's connected to a carbon that's connected to a double bond O that's connected to hydrogen, another carbon, etc., right? Everybody okay with etc.? Do I need to draw those out? Do you see that this oxygen here used to be this oxygen here? It's now connecting the two. And then this double bond O kind of twisted, spinned, and there's a double bond O, it's still there. See the double bond O's here, it's still there?
I thought you took that oxygen and connected, not the other.
You want to take the green one? You said you wanted the one here. You said you wanted this one gone.
Yeah.
Did you want this one gone?
No.
So it's still there.
Yeah, but it's on the side of the double one, isn't it?
No, the double bond's still there. It's still double bonded.
Oh, the double one just shifted.
It just shifted, just turned, just twisted. It's all very three-dimensional. I have some models in the back this week. I want you to build them. You're going to play around with them. You're going to connect them and disconnect them. Mr. Dugan brought the models in for us. They're his copies, so we're going to take care of them. And we're going to build these models so you can see how they, when you twist them, they can have different shapes and different angles. So when you're putting these together, you have to think, how do they twist? How do they turn? Yeah? Question?
How many carbons is on the chain?
On this chain, depends on the fatty acid. It can be 20s, it can be 10, it can be 5.
So what are we writing for now?
For what?
Like for now, what are we?
Whatever you want. Doesn't matter. That's not the point. How many carbons in the chain is not the point. The point is that the OH and an H leave to form what? Please pay attention. The OH and the H leave to form what? Water. H2O. Water. And then these two are connected. This is called dehydration. Do you see why it's called dehydration? And then synthesis, because we're making something.
Why is it called dehydration?
Because you're taking water out.
Okay.
Dehydration. And then synthesis because you're making something. So you could do this. You could make 3 of them and call that a triglyceride. Do you see all 3 of them there? See the double bond O? There's a double bond O. There's the oxygen connecting the chain to the glycerol molecule. There's a double bond O. It's the hydrocarbon chains there. This is a triglyceride, and you can measure how much of this is in your blood, and it tells it's one of the markers that doctors and nurses use to tell if you're healthy. Are we all good with that? No, you can't go. Are you good? So another way, oh wait, I forgot. I know where you're going. I forgot where you're going. Yes, of course you can go. Yeah, here's your hall pass. I thought you were asking to go to the bathroom. I'm used to all day someone asks me to go to the bathroom. I'm like, where is he going? All right. Next, but you don't have to put 3 fatty acids. Are you ready? Because I'm gonna erase all this. Is this okay? Can I erase? So you don't have to put 3 fatty acids. You're going to do the same process. You're going to add 2 fatty acids. So that gives you 2 waters. Notice I'm counting how many bonds I have. If you do that repeatedly, when you get to college, your life will be so much easier. Count how many bonds. Something I did not do when I was in school. So we're going to take and do it again over here. What we just did, we're going to do it over here. And you're going to stick another one on there. You see that? There's two chains. But instead of a third chain, what I'm going to do is I'm going to take a phosphate, and it's actually more complicated than this. I'm just not going to draw the big old complicated piece. There's nitrogen, there's carbons, there's all kinds of stuff. I'm going to do instead, I'm going to put a big P for phosphate. So pay attention, because you don't have to know a lot of this detail. It's really better if you just understand. I'm going to get rid of an OH and an H. I'm going to get rid of an OH and an H. I'm going to be left with hydrogen there, hydrogen here. This is going to be connected to a phosphate, and out comes water. Dehydration synthesis, H2O, comes out, just like I did with the fatty acids. So instead of drawing all this, what we can do is just say, look, I have a phosphate head, and coming off this phosphate head, which is not really coming off the head, it's actually coming off the glycerol, I have 2 fatty acids, and I'm going to represent them like this.
Is that their hydrophobic tails?
Yeah, these are the hydrophobic tails. So when we look at this chain here of carbons, that's what this hydrophobic tail is. These 2 tails are 2 fatty acids, 2 hydrocarbons, long chains of hydrocarbons that are non-polar. So this area here is non-polar. This head is hydrophilic. It loves water. Why do we use hydrophilic instead of polar? Because it could be ionic, it would still be hydrophilic. It could be polar, it would be hydrophilic. Non-polar is still hydrophobic either way. But some things could still have some charge and still be hydrophobic if they're really long. If they're extremely long, and some of these molecules like DNA are very long, so they can have different regions. Proteins can be very long. Your hair is very long. It's many protein strands all wound together. It's a very long protein. Tens of thousands of amino acids. So in any case, you can have different regions, is my point, with different qualities. So this is a phospholipid. This is the one exception with lipids not being made mostly of carbon and hydrogen. Not carbon and oxygen. Why did I not say carbon and oxygen? Take a look at this schematic. How much oxygen is in these hydrocarbons? Not a lot. Especially if the carbons turn out to be 20 or 30 long. You count the hundreds of carbons and the thousands of hydrogens, and then you can count maybe a few oxygens. So it's mainly carbon and hydrogen. That's a hydrophilic head and a hydrophobic tail that we call phospholipids. And because of that, we can draw this thing called a membrane, a bilipid phospholipid layer with a hydrophobic center. And unlike the ninth graders, we're not going to build a membrane like we did in ninth grade, because I think you get it. I think you got it in ninth grade. I don't think we need to do it again. If you want me to repeat it, we can. I just don't think it's worth our time. I'd rather you actually build these molecules with the three-dimensional objects. So you have these phospholipid bilayers that we call membranes. And of course, these membranes are in a sphere, so they're actually a circle. I don't want to say circle, I want to say a sphere. So if we were going to draw it, it'd be a round sphere. Be a round sphere, and I can't really do that three-dimensional drawing very well, but there it is. And so, but we're going to look at a cutout of that sphere, and we're going to say, look, here you have this bilayer. It's sitting here, and the heads, the hydrophilic heads, which remember in ninth grade we colored these different colors. We said and one of the things I graded when you turned it in was I looked for did you highlight that this was hydrophilic? And I said use blue for hydrophilic. I believe I remember saying blue for hydrophilic, and then yellow for hydrophobic. Yellow because it's fatty. Fats usually tend to be white or yellow, and white is difficult to draw. So in the yellow, I said use yellow for the hydrophobic center. So this would be your hydrophobic center. It'd be nice if you had colored pencils so you can draw it all in and label it. So this would be your bilayer, your lipid bilayer. Now, one of the first things we did in the lab is we can make what's called a liposome, right? You can take, if you took a bunch of these phospholipids, a bunch of these, and drop them in water, what would happen is it would form a micelle. It'd be a sphere, but it wouldn't be two layers of lipids. It'd only be one layer. And these would have tails, the phospholipid tails, because they're still phospholipids. And just they would form something like this. We call it a micelle. I think there's an E at the end, don't quote me on that. And again, it still has that hydrophilic outer area. And it has a hydrophobic center. This is really, really great for a detergent. This makes a really great detergent. Soaps work well this way. This is actually a soap. The difference is with detergents is they have a phosphate. Well, this actually would be a, because it's a phospholipid, it would be a detergent, I guess. So you have a hydrophobic center. Oh, that hurt. Oh, no. Hydrophobic center. And a hydrophilic outer layer. So just like a cell. The reason doesn't work as a cell is you only have two options. Either you're hydrophilic or you're hydrophobic. That's all you got there. Look at it. It's inside's hydrophilic, outside's hydrophobic. That's it. Now, if we did the same thing here, and again, I don't want to make a giant circle because it just would take too long. If we thought about this going all the way around, if we had this cell going all the way around, now with a bilayer, this works. Now life can exist. It's likely that the very first structure that was made, again, not getting into whether something or someone made it or not, I don't care. But it's likely when we're talking about how things work, this is likely the first thing that was made, and then this ended up being able to form these bilayers. Once you got a bilayer going, now you can have, on the inside, you can have hydrophilic. You can have stuff that's hydrophilic inside. And outside can be hydrophilic. So inside can be water, outside can be water. And the middle could be the separator because they don't mix well, correct? Why is this important? Because what is the solvent for life? Water. There is no solvent inside of my cell. There's only hydrophobic space. Here you have a solvent. Here's water. Now you can have chemical reactions that you need for life happening here. Here outside, chemical reactions can happen. And in between, you can have some chemical reactions happening. But here, this separates the two. So now you have an inside and an outside. Outside, inside. This allows life to happen because now you can trade energy for a reduction in entropy. The universe wants to be random. You look outside a cell, all you see is a random bunch of stuff moving, swishing around, moving in every direction. That's not alive. Is it moving? Yes. You can look under a microscope and see Brownian movement. You can see the movement. They're not alive. Nothing in there is alive until you add life. Life needs to be able to trade energy, and I forgot a G, energy for a reduction in entropy. In order to allow life to exist, you have to have a space that's separate from the rest of the universe. The universe is going insane. It's going in every direction. There's chaos everywhere. Life is order. Life is order. When you look at any life, what makes it different from everything around it is life is order. No matter what form it takes, a mushroom, a tree, even my R. She's order compared. Nothing, I'm just having fun. Does that make sense? So you're taking energy on planet Earth. In most forms on planet Earth, that means it's from the sun. So you're taking energy from the sun and you're trading it. You're trading it for reducing entropy. You start to put things away in spaces. You start to create order using energy. So is this understandable? Is this something that is reasonable? All right. I feel like you guys have had enough for a day. No, we're not taking time off. Don't even look at me like that. I believe you have chapter reading to complete. Yes? I believe it's due this week. 3 chapters due. I know you may not have your book with you. Or is it next week? It's next week. I'm sorry, I keep confusing the weeks. It's next week. You have 3 chapters to complete. If you don't have your book, you have Khan Academy. You can complete. You have a whole unit. You saw how many assignments were due. I promised you I'd give you time in class to get Khan done. This is your time to get Khan done. I think this was good enough for one day. Let's go over before we end the day because we like to summarize. We like Cornell notes, so let's do Cornell notes. We love Cornell notes. Cornell notes, Cornell. Give me some Cornell notes with milk. That could be breakfast. All right, so anyways, let's go review what we went over today. We started with carbon makes 4 bonds, water's polar, water's important, like dissolves like. These are big ideas. We came out with an example of a salt in a human system as urea, that ammonia, it's charged, it's an ion, needs to be made into a salt so that it can be dissolved in water, so that if it is went and then it can be excreted by the kidneys, it can be filtered by the kidneys and then excreted by the bladder and the urethra as urine. If we cannot get rid of it well, then it can form crystals and form gout, which is a disease. That's just an example of how we treat salts. Salts are an example of a solute. Salt is an example of a solute that dissolves in water. What does dissolve mean? It's a physical change, not a chemical change. It goes into solution. This when something's going into solution, which means it's being mixed well. Solute, solvent, solution. Defined what each of those were. We looked at water, and we said, look, in a cell, we have water. It's a solvent of life. Somebody says you have cell walls. I said, okay, that's true, but we really want to, right now, worry about the cell membrane. Cell membrane is made of phospholipids. I said, first let's look at phosphate. This is phosphate. By the way, it's a mineral. It's found in the earth. It's literally made, it's a rock. We are made, a big part of us is made of the earth. This is phosphate. And we have this molecule called glycerol. 2 major molecules that we said we better know. And glycerol is a type of alcohol. We said alcohols are species of chemicals that have OHs on them. Glycerol is one of those really important molecules. They said we have OH is one of those functional groups that we need to know. So there's 2 functional groups we talked about today. Hydroxide and carboxylic acid. OH, hydroxide, C double bond O, OH, carboxylic acids. We said we have these things called hydrocarbons, which make up our types of lipids. Lipids are types of hydrocarbons. They are fatty acids. Lipids can be fatty acids. Lipids can be steroids. Lipids can be fat. When we look at fatty acids, you can take a fatty acid, 3 of them, and mix them with glycerol and create something called triglyceride. And you do that using dehydration synthesis. We just take OH out and move the oxygen bonds around. If we do all three of them, they're triglyceride. If we add a phosphate instead, it would be a phospholipid. Phospholipid's interesting because there's a nonpolar end and a polar end. That lets something like a micelle form. A micelle has a hydrophilic outer end and hydrophobic center. It's a sphere. It's not a cutout like this, it's a sphere. It's round, I just can't really draw it in 3 dimensions. That's no good for life because it only gives you inside hydrophobic, outside hydrophilic, and life's solvent is water. No water can exist in here easily. So that's really not useful for life. So life needs a bilayer, 2 layers of phospholipids. That creates an outside and an inside. Huh? So I'm moving things around. So inside and outside, that allows us now that we have an inside and outside to trade energy for a reduction in entropy. Now we can have order. Now we can have life. This is why the cell is a basic unit of life, or one of the reasons, and why viruses are not considered to be alive, because they're not cells. Or a lot of people don't consider them alive. Some people are still arguing that it should be about, I don't know what percentage of the scientists are going to agree. Eventually it might be considered alive and just one of those special cases. But right now they're not alive because they don't do all the characteristics of life, which by the way, you should know what those are, right? From your chapter reading the characteristics of life. You don't need me, I don't want to talk anymore. Do you guys okay? I could talk more. I have no problem talking more, but I can see your faces. I'm just trying to summarize this stuff for you, trying to make life easier. Yeah. All right, there you go.